Okay, so welcome to this next video in which we are discussing shear stress and endothelium derived relaxation factor. Okay, so in this video what we want to look at is some of the other pathways that are involved in sensing shear stress. So far we've looked at this TRPV4 pathway where shear stress, if you get an increase in shear stress on the walls of the endothelial cells, that will activate the TRPV4 mechanoreceptor, which will then allow calcium to move into the cytoplasm of the endothelial cell through the TRPV4 channel. And that calcium will bind to calmodulin, and the calcium-calmodulin complexes will then activate the eNOS enzyme, which will start producing nitric oxide. What we now want to look at is other pathways involved in this response to an increase in shear stress. And really it's fair to say that these other pathways are just about facilitating this major pathway. So let's have a look at them. So in our endothelial cells, so if I draw an endothelial cell here, so let's say this is an endothelial cell. In the endothelial cells, you have a molecule in the membrane of endothelial cells. So I'll draw it nice and big here. And this molecule is often abbreviated to PCAM1. Okay, so let me tell you what PCAM1 stands for. So PCAM1 stands for platelet endothelial cell adhesion molecule 1. So where should I write this down? I'll write it down here. Platelet endothelium cell adhesion molecule 1. Cell adhesion molecule 1. PCAM1 for short, just taking the initials of all of those words. So I will colour PCAM1 in blue. Now, what is believed to happen is that PCAM1 somehow gets, well actually this is known to happen. What happens is that in response to an increase in shear stress, and I'm afraid this is where the pathways are all going to get awfully holy. So in response to an increase in shear stress, somehow the tyrosine residues of PCAM1 get phosphorylated. Okay, so let me go over the page and tell you about tyrosine. So tyrosine then, let me remind you of the amino acid structure of tyrosine. So tyrosine is an amino acid. Here's the amino group. So I'll start by drawing out the generic amino acid structure. Here's the alpha carbon in the center. Here's the carboxylic acid group down here, and the hydrogen off the alpha carbon. Then the R group of tyrosine is you have a methylene group here, bound to a benzene ring in the middle, like so. Okay, so here's your benzene ring, and then you have a hydroxyl group coming off it. Now, this hydroxyl group can be phosphorylated. So if I draw the structure of a phosphate group here, a phosphate group consists of a phosphorus atom double bonded to an oxygen, and then with two hydroxyl groups coming off it, like so. And then also with a single bond to another oxygen, and this other oxygen has acquired a second electron via an ionic interaction with some other chemical species, so it has a negative charge. Okay, so what's going to happen is you can form phosphoester links between this phosphate group and this hydroxyl group, this alcohol group on the benzene ring. So you can remove the hydrogen from the hydroxyl group on the benzene ring and the hydroxyl group totally from the phosphate group, like so. So let me colour this in. So you can remove this, and you can bind this hydroxyl group to this hydrogen to form water. That's why the formation of these phospho or phosphate ester links is often known as a condensation reaction. So this is a condensation reaction. Okay, because it produces condensation, basically. It produces water. Okay, now what you will then do is you will link this oxygen to the phosphorus atom of the phosphate group here, and that's now a phosphoester or a phosphate ester bond. So phosphoester is the term that's being coined because it's nice and easy to say, but the more correct term is to call it a phosphate ester bond. So this is a phosphate ester bond. Okay, right. So, what you can do is you can phosphorylate tyrosine residues by forming these phosphoester bonds between phosphate groups and the alcohol group of the tyrosine residues. And basically, what we have found is that in response to an increase in shear stress on the endothelial cells, this protein, PCAM1, platelet endothelium cell adhesion molecule 1, here, PCAM1, it gets its tyrosine residues phosphorylated. We do not know the pathway by which this occurs. So there is a sort of gap, a gaping hole, basically, in our explanation. So the single letter amino acid code for tyrosine is to use Y. Okay, so I'll show a Y residue with a phosphate group stuck on it, which I'll just denote in pink here. So this pink ball represents a phosphate group. This Y just means a tyrosine residue, so tyrosine, and the single letter amino acid code for tyrosine is Y. Okay, right, so PCAM1 somehow gets itself phosphorylated in response to an increase in hemodynamic shear stress. Okay, so this leads to this. Right, now there's another great big hole. Somehow, PCAM1 then leads to the phosphorylation of the eNOS enzyme. So here is eNOS, the endothelial nitric oxide synthase, or the NOS3 enzyme. It somehow phosphorylates eNOS, and it phosphorylates it on a serine residue. Okay, so eNOS. So let me talk to you about the structure of serine then. So serine is again an amino acid. So here's the amino group. Here's an alpha carbon with a hydrogen here. And then you have the carboxylic acid group down here. Okay, the methylene group here. And then a hydroxyl group off here. So this is the R group of serine. And again, you can add phosphate groups onto it. So eNOS will have serine residues in it, which can be phosphorylated as well. So we're going to add phosphate groups onto eNOS. Now, PCAM1 somehow leads to the phosphorylation of eNOS when it itself has been phosphorylated on tyrosine residues. We do not know how this happens. We do not know what the intermediate steps in this reaction are. We don't think PCAM1 is a kinase itself, so it's activating other things in between, basically, to cause this, but we don't know the components of that signaling pathway yet. However, adding this phosphate group on here is going to activate the eNOS. So if eNOS has been activated by calcium calmodulin complexes through the first pathway I've shown you, then this phosphorylation is going to further activate it, basically, so that both contributing together to increase the production of nitric oxide synthase. Now, how do we know that PCAM1 is involved in this? Well, basically, what you can do is you can knock out PCAM1 from endothelial cells. You can get some HUVEC cells, and you can knock PCAM1 out. You can stop them from expressing PCAM1. And I'm going to go into a little bit of detail on how you do this. So, what you do is you go to a maternity unit, you get your HUVEC cells, your human umbilical vein endothelial cells, and you grow them on a plate here. Now, those endothelial cells are perfectly normal, happy endothelial cells, so they are producing PCAM1, platelet endothelial cell adhesion molecule 1. Okay, how do you stop them producing it? Well, what you do is you use a technique known as siRNA knockout or siRNA silencing, okay? Which is a mechanism to stop cells actually making the gene product associated with certain genes. Okay, so let me explain to you what an siRNA is. So an siRNA stands for small interfering RNA. Okay, and basically they are a piece of double-stranded RNA. So if you remember back to the central dogma of biology, DNA is transcribed into RNA. That mRNA is single-stranded, so I should actually draw this out because it is actually important. So the central dogma of biology then is that in the nucleus of the cell, you have your DNA, which is a double-stranded deoxyribonucleic acid. Okay, the DNA cannot leave the nucleus. Okay, so if you want to make proteins, the protein machinery for making proteins is in the cytoplasm. So what you have to do is you have to make a copy of the genetic code for making the proteins, and this is in a piece of single-stranded mRNA. So let me move this over. So this is the central dogma of biology that I'm talking about now. Here's mRNA, here's DNA. That mRNA, standing for messenger RNA, is then translated by ribosomes. So here's a ribosome with our piece of mRNA, and I should get some colour on here to colour code this. So this is in orange, this is our piece of mRNA, and here is our piece of mRNA going through this ribosome here. And what you produce is a protein coming out here, a polypeptide, a polymer of amino acids, and this is in blue here coming down. Okay, right. So that's the process of translation. Right, so I think we'll cut it there for this video and continue our discussion of siRNAs in the next video.